Welcome to MedTechISO. I am your host Arvind Rathore, and today we will explore a real world case study on achieving ISO 1099331 compliance for implantable medical devices. Implantable devices pose unique challenges as they remain in contact with the human body for long durations, requiring rigorous evaluations for safety. In this video, I will share examples of successful compliance, the challenges these manufacturers faced, and the lessons they learned. Let us dive in. Let us first discuss why implantable devices require rigorous compliance. Implantable devices like pacemakers, orthopedic implants, and vascular stents fall under the category of permanent exposure in ISO 1099331. This means they are in contact with the body tissues or fluids for more than 30 days, necessitating a comprehensive evaluation of their biological safety. These evaluations must address both immediate risks, such as cytotoxicity and irritation, and long term concerns, including genotoxicity, carcinogenicity, and systemic toxicity. A major challenge with implantable devices is their exposure to the human body's complex biological environment. Materials can degrade, release leachables, or interact with surrounding tissues over time. This complexity makes achieving compliance a multi step process requiring detailed material analysis, biological testing, and iterative risk assessment. Let us first discuss about orthopedic implants. An orthopedic implant manufacturer working on a titanium based hip replacement faced challenges related to degradation risk. Titanium is widely used for implants due to its biocompatibility and corrosion resistance. However, under mechanical stress, it can release nanoparticles or ions over time. To address this, a manufacturer started with a comprehensive material characterization using techniques like ICP-MS to detect trace elements and stimulate wear and tear conditions. This analysis helped identify potential degradation products that might leach into surrounding tissues. Based on these findings, the team conducted biological tests, including cytotoxicity and systemic toxicity evaluations, to assess the risk posed by these leachables. To mitigate the risks, they applied a biocompatible coating to the implant's surface, which significantly reduced ion release while maintaining the device's mechanical properties. This adjustment not only ensured compliance but also improved the long term safety of the implant. So, the lesson learned here is even biocompatible materials like titanium needs application specific evaluations. Simulating real world conditions and applying surface treatments can significantly mitigate risks without compromising functionality. Now, let us discuss case 2, drug-eluting stents. Drug-eluting stents, which combine a metallic scaffolds with the drug coating, posed unique challenges for compliance. While the scaffolds required hemocompatibility testing, the drug coating added complexity, necessitating evaluations for both biological and pharmacological safety. The manufacturer conducted chemical characterization to analyze potential leachables from both the scaffold and the drug coating. Using GCMS and HPLC, they identified chemicals that could migrate into the bloodstream under simulated physiological conditions. This data informed their testing strategy, which included hemocompatibility tests for the scaffold and cytotoxicity and local toxicity tests for the drug coating. The team worked closely with toxicologists and pharmacologists to integrate the drug's therapeutic profile with its biocompatibility evaluations. This multidisciplinary approach ensured the drug remained effective while posing no biological risk over the device's lifespan. So, the lesson learned here is combination products require collaboration across disciplines. Thorough extractables and reachables studies are essential to understanding interactions between the device and its active components. Regulatory alignments is also key to ensuring smooth approval processes. Now, here is the third case study about silicone based breast implants. Silicone based breast implants present a risk of sensitization and irritation due to potential additives, and processing residues. While silicone itself has a long history of medical use, these risks can be amplified in implantable devices with prolonged exposure. To address this, the manufacturer performed a detailed chemical characterization focusing on additives and byproducts introduced during manufacturing. Accelerated aging studies helped identify degradation products that could form over time in the body. Based on the findings, the team conducted sensitization, irritation, and chronic toxicity tests to evaluate the material's long term interactions with the tissues. When a sensitizer was identified in the manufacturing process, the team modified production methods to eliminate the substance, ensuring the material's safety. This proactive approach not only ensured compliance but also improved the device's overall quality. So, the lesson learned here is manufacturing processes can introduce hidden risk, making regular audits and chemical analysis essential. Accelerated aging studies are invaluable for identifying long term risk, and iterative processes improvements often lead to safer devices. From these case studies, several key takeaways emerge that can guide your own compliance journey. First, always start with a comprehensive material characterization to understand the chemical and physical properties of your device. This foundational step helps you predict and address risk early in the process. Second, tailor your biological testing to your device's unique use case, focusing on relevant risk rather than adapting on one size fits all approach. Third, always simulate real world conditions during your evaluations. Implantable devices face dynamic biological environments, and testing under these conditions provides a more accurate assessment of long term safety. Fourth, collaboration is crucial. Work with toxicologists, material scientists, and regulatory specialists to address complex challenges and ensure that your compliance strategy is robust. Finally, document everything. A well prepared biological evaluation report BER is critical for regulatory approval and demonstrates your commitment to the patient's safety. Achieving ISO 109931 compliance for implantable devices is a challenge, but a rewarding process. These case studies highlight how real world manufacturers successfully navigated complex biological and regulatory landscapes to create safe, effective devices. By focusing on material characterization, targeted testing, and iterative improvements, you can build a strong compliance strategy that protects both patients and your organization. I hope this video has provided valuable insights into ISO 109931 compliance for implantable devices. If you have any questions or would like me to cover specific challenges, let me know in the comments below. Don't forget to like, share, and subscribe to MedTechISO for more case studies, insights, and tips on ISO standards. Thank you for watching, and I will see you in the next